Bismillahirrahmanirrahim. So this is biotechnology B and we are going to talk about DNA extraction and polymerase chain reaction. So DNA is needed to be extracted from its cell in which it is intact inside a nuclear membrane. So it is extracted by using certain detergents such as SDS, that is sodium dodecyl sulfate, and also it can be extracted by using certain enzymes. So these things are available in market and we can purchase them by naming the particular kit such as the Kaijin, and there are so many kits available in the market. So we can make use of any of them uh, based on the requirement and based on the budget as well. So, the procedure of DNA extraction begins with the lysis of the cell by using lysis buffer. This buffer actually lyses the cell so that the nucleus can come out, and also it lyses the nuclear membrane because nuclear membrane and the cell membrane are made up of lipid bilayer membrane, right? So it directly degrades that particular cell membrane so that DNA can come out. Then we go for the binding of the DNA by using the proteinase K and the wash buffer. It actually binds the DNA along with the membrane that is present in the column tube. This is a column tube. This is a ependorphin. Ependorphin is further replaced by a column tube, and column tube contains a membrane that is used for the attachment of the DNA so that each and every uh, impurity, including the protein, the lipids, and the salts, can be collected in this collection tube, right? So, after the procedure of binding, we spin it by using a centrifuge and then we wash it with buffers. Normally, washing can be taken place by using two kinds of wash buffers. One has higher concentration of alcohol and another has lower concentration of alcohol. Instead, it has higher concentration of salts. It is also used for washing out the impurities rather than the DNA from the entire solution mixture. And at each step, we go for the centrifugation. So after washing, finally we wash the column tube by using elution buffer or either the PCR grade water so that your extracted DNA can be settled into the palate of the ependorf, and hence at this step the DNA is ready to use for PCR. So these are the components of PCR, the DNA what you have previously extracted out through the entire protocol. The DNA polymerase, it is also available in market, and DNA with primers. So you get the primers from the market according to what sequence of DNA you want to identify or amplify, right? The DNTPs. The DNTPs are the molecules that are used for synthesizing or polymerizing the newly synthesizing or the daughter DNA strands after the primer. So DNA polymerase utilizes them to formulate the entire daughter strand of the DNA. And the PCR buffers. So the PCR buffers are particularly utilized for maintaining the pH level. And these are the thermocycler machines. This is the qPCR machine, and this is a simple PCR that is used for the conventional purposes. And this is droplet digital PCR system. So according to your requirement, you can make use of any of them. Furthermore, there are some PCR steps. These are the general ones, like being irrespective to the fact which type of PCR and which type of thermal cycler you are going to use. So you will be having the extracted DNA, then you will add on each and every component what we have recently discussed in the previous slide. And when the PCR will begin, so it will begin after this step, right? This is the denaturation from which the as you can see right here, this is the DNA templates. It has double helix, so it will become a single stranded one, and each of them will be annealed by using certain specific primers, right? And afterwards extension will be begin, and extension will be begin through the DNTPs and the DNA polymerase. Further, in the endpoint analysis, we are having two kinds of analysis right here. So this is the conventional one in which we get the amplicons, and we go for the gel electrophoresis to check or confirm the amplification of your particular DNA, right? Or else if you are working on a PCR that is not conventional but it is the real time one. So in the real time PCR you get such kind of graph, and you don't need to confirm the amplification of your DNA through such a gel electrophoresis, right? So basically there are multiple types of PCR, but in today's lecture we are going to talk about the real-time PCR. So real-time PCR is also of two types, that is the Tatman probe based and the CyberGreen one assay. In the CyberGreen one assay we have this CyberGreen dye that is labeled as the green color. This is the primer that primes or anneals um, the polymerization of your DNA or the amplification of your DNA, right? And this is the polymerase enzyme. So the principle of both of the types of quantitative PCR assays is the same, that after the annealing, the polymerase enzyme will begin for its extension, right? And whenever it encounters a cybergreen dye right here, so it glows up, ठीक है? It glows up and it emits light that is particularly captured by the light detectors that are already present inside the thermal cycler. But whenever we talk about the Tachman assay, here comes a probe that is highly specific in its nature because it is uh, oligonucleotide. However, the cybergreen is just a dye, it's not any sort of oligonucleotide, hence it can be attached anywhere inside the DNA sequences. It is a nucleotide specific dye, but it doesn't have any specific particular sequence where it could bind. In short, we can say it's not complementary to anything. It's just that it can attach wherever it finds the nucleotide. Either it is the right one or it is some kind of mutant nucleotide. It just attaches itself to it. However, when we talk about the Tachman probe, so it is highly specific in its nature because it is complementary to a particular sequence, right? So after the denaturation and annealing, what happens when the polymerase enzyme begins its extension activity? So the probe, because the Tachman probe is complementary, so at a specific sequence that is present on the targeted DNA, it binds itself. And when the polymerase enzyme finds it, that is already bound to its way for extension. So it cleaves it because it has an exonuclease activity as well, except for extension activity. So it binds it and it separates apart to its reporter and the quencher regions, and the quencher finally leaves the reported sequence Afterwards, it emits photons, and these photons are detected in the detector that are present in the thermal cycler. So, hence, it is more specific in its nature. However, the cyber green is, lesser specific. This is the reason that the Techman Probe assay is more reliable and more expensive than the cyber green, because it gives concise results. So thanks for watching.